I'm Kristi Miller. I'm a scientist for Fisheries and Oceans Canada. I live in British Columbia and I work at the Pacific Biological Station on Vancouver Island. I've been working for DFO for 25 years now and I run the molecular genetics laboratory. So I'm here as the lead for the environmental DNA project. Environmental DNA is basically the DNA that comes from the community of organisms living in the environment where you're sampling. So if you look at the marine environment, there are a wide diversity of organisms there. And all of those organisms are constantly shedding cells into the environment, just like a human might shed dead skin or hair. And those cells get shed into the water column, and a lot of them float up to the surface of the water. So we're able to actually take just a 2-liter sample from the surface of the water and collect the genetic composition of the community of organisms that live in that ecosystem. Today we took 5 replicate samples out of Halifax Harbour, and we're going to be taking those back into the lab and filtering them through 0.22 micron filters. And 0.22 micron filters are such a small pore size that you can even capture the bacteria. So our project is looking at multiple trophic layers. We're characterizing bacterial communities, invertebrate communities, fish communities, and marine mammal communities. What's unique about this project is that we are looking at biodiversity at multiple different trophic levels from coast to coast to coast over Canada's 3 oceans. And one of the things that we're very interested in is has biodiversity changed with global climate change, and relating to incursions of Atlantic Ocean species into the Arctic as well as Pacific Ocean species. So species that would normally live in warmer waters than the Arctic waters, now that the Arctic waters are warming, those species may be moving up into the Arctic, and they could be displacing Arctic species as well. Initially, the project started very small. When I proposed it was going to be fish, and it was my lab. And it has ballooned, and we've built, through the Canada C3, a whole new collaborative team that has actually, many of us have not worked together before, so it's pretty exciting to work with new people. This experience with the C3 expedition program has been probably one of the highlights of my 25-year career. And it's very thought-provoking. There's some really excellent science, but there's also some really excellent discussions about Canada. I'm just awed by some of the things that I'm learning.